Anna, thank you for being here. The floor is yours.
Thank you, and thank you to the organizers for inviting me and giving me the opportunity to speak today. So I come from the domain of computer vision, which has been mentioned several times today as the place where deep learning rules the ball. So today I want to tell you a little bit about how we actually use the deep learning methods and apply them to biological images, and where we think that could actually go to more than images. And to begin with, so we all know that the neural networks have made their triumphal comeback in 2012 with AlexNet winning the ImageNet image classification challenge. And since then, it was tremendous progress across all the domains of computer vision. And pretty much everywhere where we measure state-of-the-art, deep learning methods are now state-of-the-art. And this is an important caveat, because to measure what state-of-the-art is, you actually have to have a publicly accessible data set where people have labeled what they were looking for in the algorithm centers. And for natural data, this is kind of, this is simple, although for a very big data set, it still takes a lot of resources. For biological data, this is much harder. But this is actually what deep learning lives off, so we'll have to find a way of how to bring that in. So at the same time as the natural image progress has been happening, on biological images in the same year, 2012, Dan Sirisan from the Jürgen Schmidhuber lab has applied a convolutional neural network to win the 2D EM segmentation challenge at ISB, also with a very large margin. And since then, similar to the natural image world, it has been unstoppable progress everywhere for very many different tasks. But unfortunately, since the publicly accessible data sets are not as present as in the natural image world, it's hard to say if it's already everywhere. Is there actually a big difference between the natural images and the biological images that we are looking at? Because if you think about it, if you look at the image classification problems of the ImageNet style, and where you're looking if it's a cat or a dog, or if you're trying to predict in what part of the cycle the cell is, it's still an image classification problem. And at the same time, if you're trying to perform semantic segmentation and assign each pixel to a class, you can do it on natural images, you can also do it in biological images. So actually, the two big difference points is that for biological data, training data is sparse and hard to acquire. Right? You can't just put it off to Mechanical Turk or CAPTCHAs. You actually have to know what you're looking for in the images. It needs experts to produce training data. And well, another point is that, especially for medical imaging, interpretability is also quite important. And then there are also interesting technical points which make it different, like the context has a different meaning, scale has a different, is at least a little bit different. But generally speaking, it is still fairly similar, and the biological image analysis world feeds off very strongly from all the progress that we see in the natural images. So there have been a lot of effort, also in this community, for imaging single cells and for creating atlases of subcellular localization of things. It's all been really interesting, but this is actually not what I want to talk about, because this is sort of not my domain expertise. What I want to talk about is imaging cells and tissue, and finding where they are and segmenting them out of this and then doing things with them. So if you have cells and tissue, then usually if you want to segment something, then either you want to segment nuclei, if you can't segment the cells themselves, or the cells. And the nuclei segmentation is, let's say, for the standards of biological images, a well-studied problem with a lot of training data. I think because it's just a problem that is important for medical imaging applications. So just last year, we had two big challenges, one in MICI for H&D staining, exactly for the problem of nuclei segmentation, and another very interesting one that Ann Carpenter from the Broad Institute has run in Kaggle. That was a really big thing for segmenting nuclei across very different modalities. It's been widely publicized, very big money prize. It has drawn 18,000 participants, which is unheard of for images of this kind of application, 68,000 algorithms. And well, if you want to look at who actually is winning in there, so the first 4 places were all UNet modifications, and the 5th is the Mascar CNN based. So without going into any details of what that actually means, let me just say that Mascar CNN is perhaps the most popular method of segmenting objects in natural images. And the UNet is a network that was invented by Olaf Ronenberger back in 2015, and it has been more or less ruling the biological segmentation world since then. And well, in the deep learning world, 2015 is prehistoric times. So having a network that has been holding up so well shows that it really captures something about the structure of the problem that we are looking at. So I want to show you this interesting plot from the UNet review paper, which sort of shows the different applications. And an interesting thing that they found there is that if you're trying to segment from all these different modalities up there, which look, well, somewhat similar, they're all grayscale, but at the same time, they are quite different. Right? If you compare the blue and the black in there, you see that they're most of the time almost equal. And what they actually correspond to is training a network just on one modality or training on all and having a common network that just works everywhere. Right? And as you can see, just training a common network also works. Right? So for these modalities that, I guess, to a network looks sufficiently similar, you can just train one and be done with this. So this is, I think, very reassuring. But there is still the one modality for which they also train separately and for which they really put in, also in this paper, everything they can to train a really nice network. And this particular modality or this particular segmentation problem is also one that I've been working on before. And this is the problem of segmenting neurons in large stacks of EM images. And so this problem has been around for a while because neuroscientists who actually care about having it solved have realized a long time ago that they need automation and they cannot just do it manually. So they started collaborating with computer scientists, say, more than 10 years ago. And by now, it's actually almost automated. And so it's from the computer vision point of view an interesting problem, and obviously from the neuroscience point of view as well, but it's also a very good test case for cell segmentation in tissue because the objects are 3D, the shapes are very complex, they extend through the whole data set, right? Single pixel errors can be just catastrophic, right? You have anisotropic resolution if it's slice-based imaging. You have a lot of imaging artifacts, you have missing slices, you have all kinds of defects that can really throw off your segmentation. And also, the data itself is very difficult. So if you look at this zoom-in thing here, right, you see that the staining is actually not just for membranes, it stains everything. And out of that, you want to extract only the cell membrane. So this is not a trivial learning problem. Luckily, there are multiple challenges with manual ground truth. And yeah, it has been a popular domain. And we actually have something to train on. And so because there is so much training data, there has been a lot of progress. And I just wanted to show you where the field currently stays at. And these are the two groups that can actually run this kind of algorithms at petabyte scale now. And yeah, so this one is from Google. This is a flood filling network. And yeah, so they can run very big. And then there is an alternative approach from here, from Sebastian Sion's labs. But many labs actually use that and contribute to this pipeline development. And yeah, so this one is Drosophila brain. This one is a songbird. And if you want to look into the quality of this kind of segmentations in more detail, so I can show you this image from Sebastian Sion, where you see this enormous neuron fully automatically reconstructed. And then you have the red and the yellow circles, which actually signify the errors. So if you're not in this field, it's probably hard to appreciate just how amazingly good this reconstruction is. So I can tell you that when I was starting in this field in 2012 or so, you know, if you could reconstruct like that much on this screen, you already had a paper at the top conference. So there really has been a lot of progress there. And also another interesting thing about this particular segmentation problem, and with all its challenges, is that everything we learn from that, we can then reapply to all the other segmentation problems which are based on membrane information, so basically to cell segmentation. And so if you look at the other data where people try to segment cells, such as those epithelial cells, for example, it can seem kind of tempting because the data looks simple at the first glance to just go to the old school stuff and apply thresholding or something like this. And in this benchmark that was introduced last year, they have actually compared the kind of older methods and the newer ones. And you can see where the newer methods are the ones that were carried over from the EM segmentation efforts. And then you can really see, so if you look just in XY, right, and this would be the ground truth, right, this would be the no learning, right, that would be the state-of-the-art model linear tracing with UNet beforehand. So if you just look in XY, it actually looks decent. And that's what you would normally try. But if you go through the multiple steps and then also look over time, you can see that really the learning-based one is much better. And that really pays off when you afterwards have to proofread this to derive your biological conclusions. We have also seen the same effect, yes, when I was working on the segmentation of embryos in the light sheet imaging, and where we have seen that the segmentation that we have produced with a method that was also kind of reassigned from the 3D EM work is actually better than the ground truth we have trained it on, where the ground truth was produced by the more conventional no-learning methods. Even though the data does seem simple, it's never that simple if it's long enough and if you actually want to push the imaging part to the state of the art. To summarize the segmentation part here, we can now segment complex tissue volumes into individual cells. We can do it at multi-terabyte scale. It would probably still need proofreading, but actually this kind of very large scale analysis is now feasible. So what can we do with these cells once we have them segmented? Well, we know that there are single-cell gene expression atlases for multiple species at different ages, at different states, right? And we have the spatial transcriptomics methods, which can kind of bring them all into space. Is there a way how we could also add the segmented cells into that? So here I want to very quickly show you two unpublished but fairly final stage projects, both from EMBL. In one I'm involved, in the other one not. So let's start from the other one. So this is a project from Lars Hufnagel and Pierre Niveau, who are studying the embryogenesis of a small animal, marine organism called Phalusia. It's almost transparent and highly stereotypic. So what they are doing in there is that they are imaging the development by a light sheet microscope, right? And then, since it's so nicely transparent, you can segment it really well with machine learning methods over time and actually track all the cells as it goes with the whole lineage. And then once you have this, you can pick the cells out of there one by one and run the single-cell transcriptomics on them. And well, yeah, so you begin from a segmentation, right? From there you have the cell location, you have shape descriptors, you have the cells in context, you have the lineage, right? And then from there they actually sample the single cells, right? And by a method which they call transcriptome propagation, actually then see how this assignment continues. And from this they build an atlas which can be found at this webpage called digitalembryo.org. And there you can really explore this Phalusia embryo, all the genes that were found in there, where they are located, at what development stage, everything you can think of. So that was one. The other one is actually a project I am involved in collaboration with Detlef Arendt and Yannick Schwab. And here we are trying to bring together, for the animal called Platineris dumerili, two data sets which look like that. So in particular, one is this gene expression atlas that has been measured for 200 genes, or 205, but they're constantly adding to that, and all registered to a common space. It's also spatially stereotypic. Besides that one, we also have the whole animal EM data set that was taken, that was imaged at FMI Basel, and from which we can actually segment all the cells. So the whole thing is about 8 terabytes. The worm itself is about 1.5. And yeah, so that's what we actually try to segment here. And yeah, so we started from the methods that were originally developed for connectomics. We had to adapt quite a lot because there is a lot of biological variability when you are looking at the whole animal as compared to just a piece of a brain, right? But so it was very interesting. So we can segment all the nuclei, right? We can also segment all the cells. It's about 14,000 cells here. And then you can try to put the gene expression that we have in the other atlas on top of this. So Christian Tischer from EMBL has done the original alignment of light to EM. It's fairly good, right? So it's been using muscle and neuropil segmentation to bring them together. So I would even say it's really, really good, but it's not single-cell precise. Because I don't think it can really be expected that it would be, because we are comparing an average of 25 animals to one particular specimen, which also went through EM. So yeah, but we can solve that as an assignment problem afterwards, which we know from computer science, and then calculate the costs from the pairwise distances, both in gene space and in the location space. So without further detail, it can actually all be brought together, and you can have an atlas, which we here have based on Big Data Viewer, where you can really explore it cell by cell and look at all the cell properties and add the gene expression profile at every cell. And then you can also look at them morphologically and see one by one and extract the features of all the cells like that. So what can we do if we have this? So up to now, that's existing work. And from there on, I wanted to give a little bit of an outlook, maybe. So what if we actually wanted to go on and into modeling? So people have been doing image-based modeling of cells before. So Greg Johnson at the Allen Institute has published many papers on the building a 3D integrated cell model from images of individual cells with the protein localizations. There have also been a paper recently from middle 2019 also about building representations in an unsupervised way from images, right? So actually people are working on this, and we can build representation of single cells once they are segmented. At the same time, we have heard in all the talks before me that for the other kind of data, for the single cell data, we also now have a lot of ideas of how it can be represented. And I think it could all be combined together in multimodal learning setup, like we have heard in the end of Jean-Philippe's talk. So it has been done for natural images for many different modalities that have already been brought together. It would be very interesting to try a combined representation like this also for biological data, and at this I will actually finish. So to conclude, we can now segment cells and tissues at very large scale if the data is good. Right? We can, given some kind of a spatial anchor, we can actually bring the gene expression data to it. And, well, deep learning in general allows to build models from heterogeneous input sources, so I think it would be extremely interesting to try. Thank you.
Thank you very much, Anna. Do we have a couple of quick questions? Please, go ahead. Okay. I'll bring the mic to you.
You showed some really nice examples of kind of stereotypical tissues or animals or images. So you also mentioned on one of your slides the Human Protein Atlas, which does a lot of immunostaining on tissue sections, which are maybe not as typical. So are there ways to kind of typify these type of tissues to kind of making them into kind of a cartoon-like or something resembling a stereotypical architecture of the tissue from all the individual images in individual data?
Well, I would hope so, yes, that if we find the representation, right, and if we know what it corresponds to, we could learn, yes. But it's hard to do for the very first data set. But I think that there is enough information assembled now that, yes, one could try. But it is easier when you have the spatial stereotypy. But I think we could go through, finding, like if you have a cell going through cell type and then back into, yeah.
Okay.
Additional questions? I do have one. All these segmentations that you're doing, how sensible are they to the actual original image quality, and how robust are them across the entire image, and how much cells you can do at once? So.
Well, so our segmentations are dense, right? So we always do everything at once. And in terms of the image quality, well, it is sensitive in a way, right? But we can, so for the artifacts, for the missing slices, stuff like that, we can train it to ignore them if we can actually simulate it. I mean, we have done this. So you can, pull slices out and tell it, but we have still the ground truth for this, right? So if you can simulate defects, that can actually step over. So it can learn to interpolate this kind of things.
Thank you.